Hello Year 11. This lesson is about Gregor Mendel, the godfather of genetics, and the basics of inheritance. You will need your B2.3 booklets on DNA and inheritance, so that's the most recent booklet that we are on, and you need to turn to page 19 of the booklet. So the objective today is to understand the significance of this gentleman Gregor Mendel's work. So who was Gregor Mendel? Well, here's a picture of him up in the top right hand corner. He was born in 1822 in Czechoslovakia. He was a mathematician, particularly specialising in statistics, but decided to devote his life to God. So he became a monk and worked in the monastery gardens. It was here that he started to carry out some breeding experiments using pea plants. You might have parents, grandparents who grow things like broad beans, runner beans, peas, sweet peas, in the summer perhaps in the garden or on an allotment. And he was interested particularly at looking at different characteristics of these pea plants. So he studied things like the flower colour, so we get purple, white, red, different coloured flowers from peas. Looking at the plant height, so we get dwarf plants that are much shorter and can be grown in smaller containers, as well as the taller plants. Texture and colour of both the seeds and the pods, so you can get smooth or wrinkled seeds, and the pods can either be what we call inflated, puffed up, or constricted, where you can see the actual seeds sitting inside the pods. He only ever used purebred plants. So what we mean by this is plants that had both of their versions of the gene were either dominant or recessive. So I'll go over these words and what they mean, during the course of the lesson. And he crossbred them with different features. Because of his background with maths and statistics particularly, he started counting the number of offspring, the number of baby plants that he had with different characteristics. And he was interested in recording this data and collecting particularly large data sets to make his data more reliable. And he started to find out that there were clear patterns of how the characteristics were inherited. What you have to bear in mind is that Gregor Mendel lived around about the time before microscopes. So nobody had actually seen a cell or a nucleus or knew about chromosomes or even DNA. So his ideas that he came up with about inheritance of particular features were based purely on his understanding of what he could see. So he couldn't see the detail, the genes, the DNA that we can actually start to see today. So one of his first experiments, and this is on page 19, was to do with the colour of pea plants. He took two different types of coloured peas. He took some purple peas and he took some white flowering peas. And what he noticed was when he bred these together, all of the first generation of offspring, what we call the F1, were all purple. So how do you breed peas together? Well, if you're in my classroom, if you turn around and look at the back wall, there's a large diagram of a flowering plant at the back with a bee visiting it. In most cases, in nature, plants reproduce because an insect may visit a flower, get dusted with pollen, visit another flower, and it will rub off on the sticky stigma. So there are male and female parts in a flowering plant. In a female part of a flowering plant, in the middle, the flask-shaped structure that you can see, this is the female part. It has a stigma at the top here, it has the style, and it has a swollen ovary. And inside there will be an ovule. And the ovule is the equivalent of our egg cell in animal reproduction. Around the outside of the plant, you will see, some structures that look like lollipops. And basically, these are the male structures. They are called the stamens, and they're made up of the anther, the bit on the top where the pollen is made, and the filament, which is like the stalk of the lollipop structure. So for pollination to occur, we want to get pollen to be carried from here onto the stigma. And pollination that you learnt about in year 9 either can happen by insects carrying that pollen or by wind. So Mendel had to do this himself. So he used a paintbrush to take the pollen from the anthers, this part here, and to brush it onto the stigma. That pollen grain then travelled down and joined with the nucleus, okay, of the ovule. And at that moment, we call that fertilization. That fertilized ovule turns into a seed, and then Mendel would have planted these seeds, grown up the pea plants, and to see what colour the flowers were. So this experiment that he conducted didn't take place over just a day or even a week. It would have been over many, many weeks, perhaps a couple of months, for him to see the different colours, okay, that he achieved. So in the first cross, we have our purple and our white plant. They don't make pink. They make all purple flowers. So that's the F1 generation. He then did a second experiment. He took 2 of these plants and he did what we call a back cross. A back cross is when you breed the 2 of the offspring from the first generation together. And what he noticed this time round when he counted his numbers of purple and white plants was he was getting white plants or white flowers suddenly appear. And because he was interested in maths, he did this several times to repeat for reliability, and he always found he had a 3-to-1 ratio. There were always 3 purple plants for every 1 plant. So how do we explain what was actually happening? Well, in genetics, we use letters to represent some of the characteristics, the genes, okay? So for flower colour, they have used here a letter which I'm not keen on using because it looks like the same when it's written as a capital and a lowercase. But a capital P was our version of the gene that will give us purple flowers. And our lowercase p, I'm going to do mine with like a little crook, an italic p, to really make the difference. Otherwise, you can easily get confused. This was the allele for the white version of the flowers. So for our white flowers, these 2 letters are 2 lowercase. So I'll just write them next to there so you can see. But our purple flowers, these had 2 capitals. So the purple flowers are going to produce the pollen. And pollen, remember, because it's a sex cell, a gamete, is going to have half the amount of DNA. So both of the pollen grains only have 1 copy, both capital P's. And the ovules in our white flower both had lowercase. So these are lowercase p's here. So when we join this pollen grain with this ovule, we get 1 of each. And for each one, each pollen grain and each ovule joining together, they all have 1 of each different version of the allele. So we say that all of the F1 generation flowers were all purple, but a word we use to describe this thing here, which is called their genotype. So the genotype is the combination of letters, okay, that they have, is because they have 1 of each, 1 large and 1 small, so 1 dominant, 1 recessive, we say that they are described as being heterozygous. That prefix at the word hetero means 2 different. So we've got a dominant and a recessive. Both parents, we can see, were both homozygous, but our white flower is homozygous recessive because it has 2 lowercase letters, whereas our purple flower is homozygous dominant because it has 2 capital letters. So purple flowering colours or purple coloured flowers are dominant to the recessive white. So that was the first generation, the F1. Remember what he did then? He then took 2 of these flowers and he did what we call the back cross. He crossed those together. So this time, both of our parents are going to be heterozygous. They will have 1 capital dominant allele and 1 recessive allele. So we fill in the grid and you can see how easy it is to confuse our letter P's here. So I'm going to put these little P's so that we can see them and a big P like that. So this one's 2 capital P's, definitely purple, homozygous dominant purple flower. But these 2 have 1 copy of a dominant and 1 recessive. So the dominant always overpowers the recessive, and these 2 plants will be purple. Sometimes people write the recessive allele letter before the dominant. I would write it capital first, just like you would when you're writing a sentence. It doesn't matter which way round you put it. However, this particular flower is going to inherit 2 recessive copies, and so that flower is white. Now, in the exam, they might ask you, what's the probability of getting a white flower if both parents are heterozygous like this here? You can either describe it using a fraction, a percentage, or a ratio. They might specify which way they want you to present your answer, but if they don't, you can do it anyway. So you could say the probability of having a purple flower would be 3/4, or 75%, or a 3-to-1 ratio. Probability of having white flowers is 1/4, or 25%. So all of this information is on pages 19 and 20. I want you to turn over to page 21 for me then, please. And I want you to see if you can fill in the 2 grids, we call these PUNNET squares, okay, to show the inheritance in the F1 of a dominant purple flower, homozygous dominant purple flower, and a homozygous recessive white flower. And then in the grid underneath, to fill in the F2 generation, the back cross of 2 heterozygous flowers. You then need to answer the questions on what's the ratio of purple to white flowers and try to explain why some flowers are white in this F2. So in order for them to be white, both parents would have to be carrying a recessive copy of the allele. In other words, they were heterozygous. They had one of each type. And this flower happened to inherit both recessive copies. So it wasn't just flower colour that Mendel looked at. He looked at 7 different features in pea plants, okay? And he studied 22 different varieties of peas, always using large sample sizes to make his data reliable. And what he always saw in the second generation, the F2 generation, was an approximate 3-to-1 ratio. So when he's looking at seed shape, we've got here our smooth and our wrinkled. Smooth is dominant to wrinkled, and he always saw a ratio of 2.96 to 1. So if we round that up to the whole number, that's our 3-to-1 ratio. Pod shape, remember I said that they would be inflated like this or constricted, so you could actually see the shapes of the peas, and 2.95 to 1. So rounded up, that's a 3-to-1 ratio. Flower colour, which we've just looked at, the purple to white was 3.15 to 1, so rounded to the nearest whole is 3-to-1. Seed colour, we get pale yellow seeds or green seeds, and the pale yellow is actually more common, is the dominant variety, and that's a 3-to-1 ratio. The pod colour, dominant colour of peas is generally green. Now when you go to France, you sometimes see beans being sold in the market that are kind of yellowy colour, and they are called, we've got haricot vert, which is green, vert is the French for green, and haricot jaune is yellow, so is the French for yellow, and again a 3-to-1 ratio. And here we've got our regular height pea plant, which is the dominant variety, and our dwarf pea plant, our shorter varieties that people can use then in perhaps smaller gardens, and again an approximate 3-to-1 ratio. Okay, so just so that you can see some other features he studied. So your task on page 22 is to complete the Punnett squares to show inheritance of green and yellow pea seeds using these symbols. Now, in your booklet, there's not a mistake, but in this slide, that is a mistake. That should be a lowercase. Yellow peas are represented by a capital Y, and lowercase y is our green peas. Remember that the plant needs to have 2 copies of this, okay? Just like you have 2 copies of every type of gene. One from mum, one from dad. Peas get them one from the male, one from the female plant. So we've got our yellow peas would have to be purebred, so they would be 2 capital Y's. But our green peas would have to be 2 lowercase y's, okay? The F1 generation, so when you fill it in on here, okay, they are all going to be heterozygous because they've inherited one of each version of the alleles from their parents. And then these are crossed together, and if we look, we've got 1, 2, 3, 4, 5, 6 yellows and 2 greens. And if we look at that 6:2 ratio, if we simplify it, it's actually a 3:1 ratio. 3 yellow peas for every 1 green pea. So pause the video, see if you can fill in the Punnett squares, and then when we restart it again, you will have the answers there for you. So our male alleles are going to be 2 little y's, our female alleles 2 capitals. When we fill it in, each of the offspring will have 1 of each, so we describe these offspring as being heterozygous. And then when we do the back cross, each of the parents this time are heterozygous. This offspring will be homozygous dominant, will definitely have yellow peas. This offspring will be heterozygous, okay, and have yellow peas. So will this one, but this individual here will have our green peas because it's got 2 recessive alleles. So answer the questions at the bottom of page 22 about the ratio in the F2 generation and explaining why some peas are green. Over the page, page 23, we're going to look at an example of eye colour. Before we do, I just want to go over a few things. So in all living things, characteristics are passed on in their chromosomes that the offspring inherit from their parents. Chromosomes are always matched in pairs, and 1 chromosome comes from each parent. So you might have 1 coming from the mother and 1 coming from the father, 1 from the female parent, 1 from the male. So the genes in the matching pairs, are they exactly the same? Well, even though they're at the same place, so you can see these dark blue bands opposite one another, they may not always be the same versions of the gene. So we call those different versions the alleles. So the chromosomes in a matching pair contain the same type of genes. So we're going to use the idea of petal colour. Each chromosome may have a different version of a gene. So let's say this one is for red petals on the left and the one for yellow petals on the right. The different versions of the gene are called alleles. So in our diagram, if the alleles in a matching pair are the same, they are called homozygous. So if you've got 2 yellow petals and the animation is not working currently as we open this flower, normally that would turn into yellow petals on there. And if they've got 2 red petals, if we opened this flower, it would have red petals on there. So that is our word homozygous. But if we've got 1 of each, if we've got a red allele and a yellow allele, we describe the genotype as being heterozygous. So in this case, okay, the allele for red petals will overpower the allele for yellow. So because it's dominant, okay? So the dominant form is always expressed. Letters are used to represent different alleles. So a dominant allele always has a capital letter. So if we're looking at colour of petals red and yellow, we're going to use capital R for our dominant red. And the recessive allele is always the corresponding small letter. So don't think that you would do a different letter and you would have like a Y. It has to be the lowercase letter to show the recessive allele. The pair of alleles is called the genotype. So you could have 2 capital R's would be red, 2 lowercase r's would be yellow, and 1 of each, capital R, little r, would be red because the dominant allele would overpower the recessive lowercase one. So the allele pair for each characteristic is called its genotype. The physical expression of the allele pair is the phenotype. In this case, what is the colour of the petals? So if we've got a genotype that is 2 capital R's, this would be red, So if we clicked on this bud, it would give us red flowers if it was working. This one would be yellow, 2 lowercase, and this final one, this heterozygous one, would be red 2. So we've got some definitions for you to complete on page 24. These are just a few of them. We've got a gene is a section of DNA that codes for a particular characteristic or trait. An allele is a different version of a gene that codes for a different version of a characteristic. Dominant is an allele that is always expressed even if there's only one of these present, because it will dominate the recessive copy. And recessive is an allele that will only be expressed or shown if you have both alleles are lowercase letters, okay? Because if it has a capital, it will be dominated by the dominant one. So they're just 4 of the terms. You've got to see if you can match up the terms on page 24 for me, please. So pause the video and have a go at doing that. And so all of these words that I've put here to understand what's meant by these following genetic terms, this is what you're doing currently, matching up those words, okay? So just a couple of things to remember. Chromosomes are strings of genes and they occur in pairs in each cell. There are 2 copies of each gene in each cell. The genes may be identical, so they may be homozygous, or they may be different. They may be heterozygous, okay? And we call the different versions of the genes the alleles. In our little cartoon here, our dominant B allele is overpowering the little b. So a dominant allele is one which controls the development of a characteristic when it's only present on one chromosome. You only need one copy of it. Recessive, you need to have both copies would have to be lowercase b for that feature, that characteristic to be shown. So a bit of an overview. Homozygous, 2 identical alleles for a particular characteristic. Heterozygous, 2 different alleles for a characteristic. Genotype are the letters, the alleles, and the phenotype is the actual physical appearance, the outward effect of these alleles. A couple of other definitions. The F1 is known as the first generation and the F2 is the second generation. Selfing is not taking a picture of yourself. It is manually pollinating a flower by placing its pollen on its own stigma, and it's called self-pollination. Monohybrid inheritance, which is what you're studying at GCSE, this prefix mono means one version of the gene. Hybrid, if you think of a car, you can have electric and petrol these days, okay? It means that it could be a combination of 2 different versions of that one type of feature. So it's the study of how a single gene is passed on from parents to offspring. We use a PUNNET square, which is a type of grid to predict an outcome. And your genetic crosses, your top tips when you're doing these grids is to make sure that you state what the symbols represent. Try and use a symbol, a letter, which is obviously different, written as a capital and a lowercase, so you don't get confused. Make sure you label each line in the cross with the phenotype and the genotype. Circle the gametes to show that meiosis has happened and read questions carefully. So these are some of the words that are on that page 24. Pause the video, see if you can match them up, and it will then show you the answers. So pause the video, check you've got some of those answers correct, and then do the others. Now, just as an additional little bit of work, I want you to meet Bob. Bob is very confused. He needs your help. He has blue eyes, but both of his parents have brown eyes. So how can Bob have blue eyes? Well, genes are found on chromosomes. Chromosomes come in pairs, one from each parent, and each chromosome will code for a particular characteristic. Chromosomes coding for different characteristics are known as alleles. So Bob is going to inherit 2 versions of this particular gene for eye colour from his parents, one for mum and one for dad. Some alleles will be dominant over others. So in this case, brown eyes is a dominant allele, so we give it a capital B. But other alleles will be recessive, so we will give them a lowercase b. So when the egg that made Bob was fertilised, the chromosomes coding for eye colour joined together. He could either have had brown eyes, in which case he would have inherited two dominant capital B alleles from both parents, and so he would be homozygous. He could have inherited two lowercase blue eyes and have two lowercase b's, and again be homozygous, but this time homozygous recessive. Or he could have inherited one of each type of allele, a capital B and a lowercase b. He would have brown eyes, and he would still be heterozygous. However, we know that both of his parents had brown eyes. So how is it that Bob has got blue eyes? So if Bob's dad had brown eyes and Bob's mum had brown eyes, they both had to be heterozygous in order to pass on the recessive copy. So let's just check this out. If Bob's mum and dad both had brown eyes and they were both homozygous dominant, all of the children would have brown eyes in this example. So if we fill it in, we would have capital B, 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 capital B. 100% of the children would have brown eyes. What about if Bob's mum was heterozygous for brown eyes and Bob's dad was homozygous? All of the children still would have brown eyes. However, these two would be homozygous for brown eyes, like Bob's dad, and these two would be heterozygous for brown eyes, like Bob's mum. What if it was the other way round and Bob's mum was homozygous? Well, they would still have 50% would be brown and 50% would be brown heterozygous. So these are homozygous, these are heterozygous. So 100% brown-eyed children. So the only way for Bob's mum and dad to have a blue-eyed child, if they've both got brown eyes, is they both need to be heterozygous. So we have 3/4 chance of the children being brown-eyed, but there's this slim chance, 1/4, 25%, okay, a 3-to-1 ratio, that a child would have blue eyes here. So what I would like you to do, and if we had both were blue eyes, obviously there's no chance of having a brown-eyed child. They would all have blue eyes. So what I would like you to do is on page 23, there are 2 Punnett squares for you to fill in, 2 grids, okay, to show mother has blue eyes, 2 lowercase b's, father has brown eyes. Can you fill in the first grid and say what colour the eyes the children will have? And then the second grid, should be underneath question 3. It's got pushed up. Can you draw in this grid 2 parents who are like Bob's, who are both heterozygous, and tell me what the probability of having those coloured eyes? Thank you, Year 11.